everyone, and welcome to today's webinar, Delivering Improved Bioprocessing Workflows, the Manufacturing and Science Technology MSAT Service. I'm Borga Kultz, Product Manager for Services, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by FUJIFILM Irvine Scientific. To learn more, visit irvinesci.com. We do encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like now to welcome our speakers, Tom Fletcher, Director of Process Development R&D at FUJIFILM Irvine Scientific, and Michael de la Cruz, Ph.D., Senior Scientist at FUJIFILM Irvine Scientific. Tom and Mike, you may now begin your presentation.
Yeah, thank you so much, Borga, for the introduction. So it's my pleasure today to tell you about this new service we're very excited about. So FUJIFILM, as you may know, has made some strategic investments in life sciences, and many of these investments have to do with cell biology. And so here's kind of a chart of various companies that have been acquired by FUJIFILM strategically. And so FUJIFILM Irvine Scientific is one of those companies. And what ties a lot of these companies together also is the need for cell culture media. So that's kind of our specialty. And so we're very happy to be part of this family of life science companies. It's not only good for our business, but honestly, we've learned so much since in just a few years we've been part of this family, because now we have a rich variety of collaborators, and so it's really strengthened our knowledge base. So we've been in the cell culture media business for 50 years, but we're still learning, and it's been really a great experience for us to work with some of these companies that you may recognize. Everyone from FUJIFILM Diosynth, which is a CDMO, to FUJIFILM Waka, which is a chemical supplier. So this is kind of background for who we are and why it's such a dynamic time at our company. So you may know also that we have a reputation over our many years of developing products that have been designed for the purpose of use. And so it's this design principle, again, you may be familiar with, is very common. Begin with the end in mind. So this is how we approach new product development. And so I've listed here in the bullet points some of the characteristics that go along with products that have been designed with the end in mind. So what we call purpose-built products. And so this has been, our practice, again, over many years, but recently we realized that, since we have a large business also in manufacturing media that belong to our customers, where the formula belongs to our customers, we realized it was fairly obvious that maybe we should begin offering these advantages to our customers, who own their own formula. So not just our own products, but, since we've got this experience and it's quite valuable, why don't we extend, that know-how and that capability to help our customers improve their processes. So, for the most part, this relates to practical issues. I talk about being soluble, being filterable, being stable. And then I put a star next to this one bullet point that says no pH, osmolality, or volume adjustments required. So again, we know how to do that. We've been doing it over many years. As you know, we sell both liquid and powder, and we've learned how to make those, basically equivalent, and also so that the powder can just be added to water, quickly dissolved and used. So this is sort of, in a nutshell, what the MSAT service is all about. We just ask that obvious question: how can we take these advantages that we know how to achieve and offer those to our customers who own their own formulas? And so, again, let me just expand a little bit so that you understand how relevant this is. You probably recognize some of these issues on the slide here. So there are very common issues that we find, throughout the industry with powder media in particular. And so there are some of the issues in the first group, I would say you could describe as chemical issues. So it has to do with how to hydrate the powder, whether it's solubility, interactions between the components. So kind of technical issues related to chemistry. The second group of bullet points here are more about practical issues related to the amount of labor, the amount of handling, decision making, human intervention. And of course, this is where a lot of the variation, a lot of the opportunities for errors occur when the technician is required to, do something very, very carefully. And so these are two categories. We believe we can address both of them, again, based on experience. So this is, again, kind of a general description at a high level what the MSAT service is about. We'll have some case studies here coming near the end of the presentation. But again, from a high level, we had, quite a bit of experience offering services. So we don't just sell products and we don't just manufacture custom media. We actually have a range of services. Those services until now have been focused really on early stage projects where people are developing media. We have ways to help them develop those media more rapidly, more efficiently. But as you know, what happens during drug development is as a candidate moves through clinical trials, the needs and the interests change. So as a project moves into late stages, it's not as much about speed anymore as it is about reliability and cost. And so realizing that the needs are changing and being actually requested to help as a full service media partner, our customers will call us first when there's an issue, anything related to media. So more and more we were noticing that the needs were changing. We were noticing that we didn't always have, the right service to meet the need. And so again, it came somewhat organically through interacting with our customers that we realized what we need to do is to begin offering this type of a service that would help with a more mature project in late stages, help them with practical issues of how to prepare the media at large scale, how to do it with less variation, less errors, and how to do it in a more cost-effective manner. So again, if you go all the way to the right, I list some examples of where this provides benefits, where we can improve solubility, we can troubleshoot, their bill of materials, we can help simplify their process, and we can even apply automation. So that's something I'll just mention briefly, but we know how to prepare media. We do it every single day. And so through that experience, we're able to offer, I believe, a valuable service to help our customers. And so again, just to help bring this to reality for those of you who are involved in large-scale media prep, here's some very specific examples, just a list, but it ends up being a long list. It's not so much these particular examples as how long the list is of different things that people need. So again, as a full-service media partner, we hear a lot about issues like this, and so we can certainly improve somebody's process simply by delivering ready-to-use packaging, pre-measured quantities. We know how to design the powder media so that the adjustments won't be required. There's so much time and variation being introduced by having somebody make adjustments. We know how to integrate nutrients into a single bulk powder instead of adding, each one individually. And at the same time, we know how to keep those components that might cause trouble because they interact, we can separate them. And again, I won't read the whole list, but you get the idea that just from experience and through understanding what the risks are, we're able to overcome some of these very common challenges. And obviously it's rooted in fundamental chemistry. So I'm just going to pick on one component here. It's got a reputation, and that is the tyrosine. So tyrosine causes a lot of issues in cell culture media, in feed media. And so it's, once you understand the chemistry of tyrosine, I think it helps you understand why there's not only so much issue with it, but how there are opportunities to overcome those issues. So for example, if we simply will use a high pH for dissolving tyrosine, we'll probably be able to achieve a much higher solubility and a better stability also of this amino acid. So it's unique in several ways compared to other amino acids. And so that explains, I think, why it's also unique in terms of, the amount of challenges it presents to us. So just looking at some diagrams here of how the pH influences the structure and the behavior of tyrosine. Again, we can look at this and realize, oh, well, that helps us understand how to deal with those practical challenges with tyrosine. So we might introduce it as a separate stock solution at high concentrations. And again, it's all based in fundamental chemistry. It's not that we're inventing, any novel chemistry or proprietary chemistry. It's that we're taking knowledge that's available to us and applying it to the benefit of our customer. So, similar to tyrosine, we can recognize individual nutrients and their behavior. For example, here I'm just showing the effect of sodium chloride. So the ionic strength of the solution affects solubility. In the case of cysteine, we have kind of a salting in effect. Tyrosine and leucine, just the opposite. So just knowing these things, having a knowledge base that's available to us to design media to help solve problems, I think is really where the benefit comes from. And then, by combining these effects, we realize it quickly becomes complex. And so I'm just showing you here very briefly how, looking at 2 amino acids at a time is also interesting. And thanks to not only our own work, but some of the work we've done in collaboration with groups like AMBIC, for example, we're able to understand and apply what's been learned across the industry in terms of how amino acids affect each other. So quickly on the chart at the right, I'm showing how with alanine, there are some amino acids that improve the solubility and other amino acids that actually have a negative effect. So these amino acids will impact each other in terms of the combined solubility of the amino acids. And then just very quickly, just before we get into case studies, I just want to tell you there's a bright future in this area also, which keeps us motivated and excited. We're excited to be part of the AMBIC consortium, where we're working with many other companies in the industry. And part of that effort that we're excited about is developing in silico models where we'll be able to make predictions about how to combine different nutrients, how to group the nutrients in ways that are most cost effective and most reliable. And so that's something we're quite excited about. It will really save time and I think result in more reliable media design. In the second case, we're also very excited about a new automated approach to media development. And I wish I could tell you more about it right here, but we'll certainly be talking about this more in the future. We've designed a system that's able to, based on the same chemistry, able to achieve rapid and reliable automated media and buffer prep. So that's the end of my section. Apologize if I went very fast there, but I'd like to hand it over to Mike De La Cruz for some description of actual case studies.
Thank you, Tom, for the great overview of the service. I hope that everyone can see that the MSAT service addresses the existing lack of support for the media preparation unit operation at the late stage of bioproduction. Next, I'll present a case study of an opportunity we recently worked on. As you will see, it gives a good representation of what the service is all about and what solutions it can provide to your manufacturing workflow. Our main goal is to improve the media preparation method by simplifying a complicated, multi-component, multi-powder liquid preparation into easy-to-use powders that are easy to hydrate and scalable all the way up to GMP manufacturing. So as you will see in this case, we started with 3 powders and a very complicated media preparation method that typically took 4 hours to make, and we ended with a simple 1-hour hydration process. And the real value comes from the mitigation of risk, where later you will avoid any variability introduced in the process, and you can follow a lean manufacturing methodology, which is so important now with multi-product facilities, ensuring there's no disruption to the supply chain due to any issues with the media. At the end, it is all about optimizing the efficiency of your DS manufacturing facility. In this case study, we were approached by a previous customer who had used a couple of our products, including 2 of FUJIFILM's media server panels, a growth and a feed media panel. This customer had developed a production media for a drug substance antibody process that could potentially use more than 100 kilograms per liter of media per year. Yet they knew that the preparation method was not manufacturing friendly, since it was typically taking up to 4 hours to prepare the medium each time, even at small scale. So this was going to be a real challenge at large scale. One of the main reasons for the issues encountered was that they had co-developed a supplement powder with a CDMO, which did not fully hydrate as a complete powder when it was first ordered from our express media service. But because our customers' previous experience with this powder, they understood that it would be fully solubilized when the pH was adjusted up to 9.5, and it would stay in solution when it was adjusted back to neutral pH. Therefore, the limit of solubility of the powder components had not been reached. The basic strategy that the MSAT service follow for this particular customer can be divided into four basic tasks of redesigning the formula and method, which are depicted here in this circular diagram. Starting at the top left, we first compiled and tabulated the customer's formula information. This allows us to compare the formula to both internal and other commercial media to look for any potential challenges due to solubility limits or undesirable interactions between the components. Then, in the next three steps, we turn our attention to the method, where we eliminated the complicated adjustments which can be avoided. We eliminated the pH adjustments by balancing some of the weak acids with the conjugated bases in order to meet the pH set points. At this point, we also had to be mindful that not all components will be added in the main powder. Therefore, the impact to the pH of these additions had to be better understood, which could only be done empirically. And to really simplify the preparation, the usual hard-to-dissolve components were prepared as a high pH concentrated liquid supplement. This supplement is added during the preparation and completely eliminates the need for pH titration at the point of use. We eliminated the need for any osmolality adjustments, and because the amount of titrants used to solubilize the hard-to-dissolve compounds was minimized, we were able to eliminate the issues with high osmolarity that the customer was having with the old method that made it a challenge to meet the final specifications of the liquid media. Additionally, we eliminated the need for adjusting the volume at the end since we can accurately determine how much water to start with. And as the diagram shows, this was an iterative process where these four activities were revisited as the formula and method were fully redeveloped and optimized. Our main goal for this particular case was to simplify the media preparation method in order to streamline the manufacturing process workflow. As I mentioned, the customer had used FUJIFILM growth and feed media survey panels, as well as a supplement powder not developed by us to find an optimal production media formula. Yet when combined, these powders were insoluble without a pH manipulation, making the hydration method very complicated and long. But by using designing and engineering activities known to resolve these and similar issues with media preparation, which included substituting components with equivalent nutrient compounds with higher solubility limits, which include the sodium salts of amino acids and dipeptides. We also use alternate metal complexes that avoided detrimental changes in the redox state of these components when in solution that led to precipitant formation. These and other changes allow us to combine the majority of the components into a single powder, and as I mentioned, the hard-to-dissolve components were provided to the customers as a high pH concentrated supplement solution that is added during preparation at the point of use. We and the customer understood that these changes helped increase the reliability of this unit operation by eliminating avoidable interventions that introduce variability and error to their process. This in turn reduces the amount of time and labor that is allocated to the media preparation. They save time and extra extra time could potentially be used on other activities that will help increase the overall efficiency of the facility. And because the preparation method is so simple, it's guaranteed to easily scale up and be transferred to any manufacturing facility. So at the end, we were able to provide our customer with a user-friendly media and a concentrated supplement solution to address the solubility issues encountered and a simplified preparation method. In this series of pictures, you can see how the media was prepared at small scale to verify the final formula and hydration method before sending it to the customer. After the addition of the concentrated liquid supplement, the pH shifts from 5 to just about 7, after which the solution will start to become clear and eventually becoming completely translucent. The final targeted pH of 7.5 is achieved after the sodium bicarbonate is added. Since we started with the exact volume of water that is required, no final adjustment to the volume were made. And finally, the medium was filtered and stored cold. So we quickly received positive feedback from the customer that the preparation time had been reduced down to about an hour. Here's the composition of the final single production medium powder in terms of percentage of each of the original powders used. 45% of the final powder is represented by the growth medium, 20% by the feed medium, and 35% is represented by the supplement powder developed by the customer. And we were able to provide any of our customer with highly concentrated, high pH liquid supplements, which in this case represents 1% of the final volume. And as I said, we received positive feedback from the group who had previously been preparing the media, and they were really happy to have a single powder to hydrate and found that the new simplified preparation method with minimal additions and no adjustments was easy to follow. Because this customer used Fujifilm's MSAT service, they are better prepared to adopt the recently launched automated media preparation system, the OSIO rover, since the methodologies used in both the media cartridges used in the OSIO rover and the MSAT service are based on the same basic chemistry. And what we like most to hear most is that our customers are fully satisfied with their experience using the MSAT service with positive feedback like this. These are direct quotes from our customers after they had tested the powder and preparation method in their lab, telling us how the preparation was now a wonderful experience as they had been able to complete a 10-liter preparation and filtering within 1 hour, where it would have typically taken them between 3 to 4 hours. The final pH and osmolarity set points were met, and the metabolite measurements were comparable to previous preparations. And finally, the customers confirmed that the media met the expected cell culture performance. In takeaway, we have identified customers' gaps. Phi-Sci has the resources and capabilities needed to address these gaps, and with the MSAT service offerings, we will be able to meet our customers' needs at the late stage of process development. Thank you for listening.
Thanks for that introduction to the MSAT program, Tom and Mike. We wanted to chat some more today about some of the work we've done since we started MSAT last September and some of the issues that clients have come to us with. And one thing we've been asked, since we do have several existing programs like MDO, Media Development and Optimization, and the Express Media Service, why did we also feel that we needed MSAT?
Okay, right. Well, I'd love to tell you that it was the clever brainchild, of a brilliant businessman, but that's actually not the case. What it was, so we noticed, that we were being asked to help on certain types of projects. We noticed that there was a need, and basically we were having customers ask this, And so we did recognize that it was an unmet need. And that was really the driver for us to initiate this. This service, as I think I mentioned on one of the slides, is more about, the practical issues that come later on in a drug development a timeline. So it's more around, when the change in interest comes down to practical issues or cost of goods. And so again, we're focused on really helping to streamline that media prep, that buffer prep, those types of processes. And so, I can go quickly through like the EMS service that you mentioned. That's about rapidly providing materials to the customer to support their development activities, so they don't have to wait for the full GMP manufacturing of their media. So we can quickly get those materials into their hands, in 10 days or less, I think is what we can do there. And then for spent media analysis, that's very important during media and process development, so we can do things like that. And then the MDO is like a full service media development program. So we can do all of those things. They're quite successful, but this was more focused on the later stages of drug development, where it's not as much about speed. I'm not saying that we're slow either, by the way, but it's more focused on how to save time, how to reduce labor, how to improve the efficiency of the process, and obviously how to avoid these risks that we've been talking about with solubility, stability, what I would categorize as very practical issues that have to do with scaling up and creating a robust process for commercial use.
And customers have asked us how those advantages that you mentioned earlier of purpose-built products, how those could apply to their own media?
Right. Okay. So when I mentioned purpose-built, I was talking about our own products that we sell commercially. And so as we develop those, we've just learned about how to make them easy to use, how to avoid, the intervention that's often required. And keep in mind, of course, we're manufacturing these products not only as powders, but also as liquids. And one thing that we do that I think is different than a lot of our competition, we're actually manufacturing the complete bulk powders. We sell those, of course, but we use them also to make the liquids. There's no question about comparability between the liquid and powder because of that practice. Sometimes you'll hear in the industry this phrase, the liquid is made from first principles. And I'm not sure where that phrase originated, but it means that people are making the liquid by adding one nutrient or one component at a time. So that's something we almost never do here. It's we make a complete bulk powder, then we'll make the liquid. And through that experience of understanding how to design the bulk powder so that there's no adjustments, so that it's easy to prepare as a liquid, we realized we were onto something that could benefit our customers as well. And so that's kind of where we took that purpose-built concept, we learned from it, and then as we engaged with customers and learned more about their processes, we were honestly quite surprised to see how much how much time and effort was being put into simple liquid media prep. And for us it was fairly obvious. We're like, oh well, had you ever thought about pre-engineering your powder so you don't need your operators to be so busy making adjustments? And it's really not just about the time and effort, it's about the variation that can result when you're relying on your technician to make these critical adjustments and things like that. So again, just by doing this for a long time, I would say we've kind of learned that there are smarter ways of doing this, and decided that would be valuable to our customers. And so we're excited to bring that value to our customers. It's not something we want to keep selfishly and kind of leverage it by trying to persuade people to buy our off-the-shelf media. We, I guess consistently with our philosophy of helping our customers succeed, and that's the best way for us to generate long-term success, we're out there offering this know-how, you might call it know-how, to help our customers succeed. And we're happy to offer that experience to the benefit of our customers.
Yeah, and we have several examples where we have actually applied some of these methodologies that have been developed here over the course of 50 years of preparing media. In some of these examples, we basically are focusing on, looking at the formula and the method. Sometimes we can do both, sometimes we could do just one. So, for example, in the case that I presented, I showed that we were able to modify that media formula because the customer was actually able to do so and willing to do so. So in those cases, we we were able to incorporate those 3 powders into one by exchanging some of the few components and substituting them with more soluble components. We were able to improve the solubility of that media. But we also looked at the method and tried to simplify as much as possible. We eliminated any of the pH adjustments, volume adjustments. But we have also worked with some clients that don't have that flexibility of changing their formula. They pretty much have it locked down. And what they really want to have us do is look at some particular issues they're having with, the media. So it could be, the actual, they want to see whether the actual method and factors that are involved in that method are contributing to these issues that they're observing. There could be, stability issues or issues of cell culture performance. So, for example, we worked with a client that was having an issue of stability, and they weren't able to meet their schedules because the media wasn't stable for as long as they needed to be. So we quickly were able to do a root cause analysis and identify some of the factors that were affecting the performance of the media. And, it was a very simple solution. They didn't have to do much. It was basically changing the way that they handle the powder and the way that we package the powder and provide it to them that allow us to take care of that issue very simply by, making some simple changes. In that case, it was because the powder was susceptible to moisture. So we knew that with better handling of the powder, they wouldn't run into those problems. In another case, we worked with another client that had some issues that were in the cell culture performance, and they wanted to see whether or not some of these factors in their media preparation, because they had a very, difficult and complex method where it involved pH adjustments, osmolarity adjustments, and volume adjustments. So we took those factors and identified which, one of those were actually contributing to this effect that they were seeing through by building a DOE. And so we were able to provide them with the prototypes to run those DOEs, which in this case, we had a 4-factor DOE, which allows us to do this screening with 11 prototypes, which is in about 23 runs with a center point as well. So by doing this methodology, we were actually able to identify that some of these factors, it was actually a 2-factor interaction that seemed to be very significant when pH and osmolarity were interacting, seemed to have a detrimental effect later on in the culture. Not only that, but each of those independent factors were also significant. But we also found out that, for example, the volume adjustment at the end really wasn't contributing much to that effect. So in that way, you were able to focus on those particular factors that you need to really have full control and maybe tighten the specs of your method so that you can reliably make this media without the variability that they were seeing. So yeah, I mean, the ultimate goal is to basically improve their methods of hydration so that the unit operation is more reliable and their efficiency of their facility is improved.
Yeah, and as you give those examples, Mike, it reminds me that, a lot of these projects that we've worked on, and we didn't realize this in the beginning, but they kind of come out of investigations. And so I guess another way of saying it is people don't realize that they need this kind of a service until they bump into a problem and say, can you help me? What do I do? Because they're having issues and problems. So, it's always better if we would have done this ahead of time. Hindsight is, 2020, but what ends up happening, most of these projects are generated when people do run into problems. And so it starts as an investigation, and then this service ends up being, part of the solution to those issues.
Mhm.
So I'm not saying it should always go that way, but that's been most of the work we've had so far has been kind of problem solving.
Yeah.
Well, and based on some of the questions we've gotten from clients, maybe it's important to mention that we can work both on some combination of our own FISI media formulations that customers are using, but also really extensively with clients' formulas too, can't we?
Oh yeah, absolutely. There's no restriction there. In fact, again, as we think through the examples of what we've been doing so far, I think it's mostly customer-owned formulas. So there's no obstacle there in terms of IP ownership or the formula, where we start from. It's really focused on these practical issues. And again, there's no game here, that we're trying to win an IP position or something like that. This is really just us bringing our experience to the table and helping overcome challenges or prevent challenges. I would rather than most of the projects we're preventing these kind of challenges, but we'll take the opportunities as they come and solve the problems that we can.
Yeah, so we've gone through several of these already, and we already know how the process goes. We meet with the client at the beginning, and we really like to spend most of our time in the planning stage at the beginning of that stage, so that we could clearly identify that issue that they're encountering, so that we could identify what actual factors are influencing that effect that they're seeing. And we do this through root cause analysis and so on. And through the several iterations of planning and experimental building, we come up with the actual experimental plan, which we're part of that in terms of providing you with the prototypes. And most of the times, the client is doing the screening on site. And it really depends, on the client's capabilities of how big or how they would like to conduct those experiments. So all of that is worked out through all this process of planning and then executing the experiment on site. And once they have come up with all the data, they provide that to us, and we analyze it for them, and we then are able to provide those easy to use solutions and to improve their methodologies, their hydration processes.
Since we began offering the MSAT service last September, we had the privilege of helping customers resolve numerous cases of preparation issues that attendees here might find interesting too. Could you tell us a little bit more about those challenges?
Yeah, sure. I mean, I guess we've been talking about them a little bit already, but one thing that surprised us, and I just mentioned this, is that so many of them were still trying to understand what the problem was. And so it wasn't like they had a clear understanding before we came. And so we, in many cases, it seems, will be starting with an investigation or trying to identify, kind of a root cause before we try any kind of particular solution. And so that's fine. I mean, but it is important that we first understand, what it is. Sometimes it involves recreating the problem just so we know what it is we need to address. But after that, I would say that, our solutions are generally two different varieties, kind of chemistry solutions or, more practical. Mike, you can probably tell a little bit more about that, like what kind of solutions.
Yeah, going back to our examples. So for example, the case study that I presented in the presentation, we were dealing with a powder that was actually being prepared, as you described, with first principle. And this powder not only contained free amino acids that we know that are hard to dissolve, but also some components that could be easily oxidized, like the first form of the first. And so we were able to modify that formula so that we could get rid of those components that were hard to dissolve in that way, and substitute some with the salts that are more easily to hydrate. And that way we're able to combine all the powders into one, and also resolve the issue of a possible issue of instability with the ferrous form that could easily oxidize. So we replace that with the ferric form of the compound. And another way that we've been able to also help out is by changing the method. And a very easy solution that exists out there that many people are still using, and we're very surprised that they still are, is that they're doing that last QS step, where they adjust the volume. So they're starting with about 80% of the volume typically, and then adding the powder and then doing that volume adjustment. And we think that actually could add on variability to the process. So by eliminating that and starting with a set volume, that actually can control that variance that we sometimes see. So yeah, I mean, many different ways of tackling these issues of solubility.
Another topic we encounter when we talk to our users is how improvements that we develop in MSAT could reduce the costs for their cell culture process. Could you speak to that, please?
Well, okay, so we can certainly reduce costs of their overall process, but it's typically by saving time on the media preparation part. I guess now that I think about the question, so if we can reduce variation, in the cell culture process by, having a more reliable or more consistent media prep, then I think that is definitely going to save them money. It's going to save them a lot of headaches too. So as we develop more reliable, simplified methods for media prep, and even as we design the packaging, like ready to use packaging or whatever it takes to make that process more scalable, more reliable, I think that saves a lot of money and a lot of headaches, as I mentioned.
Yeah, I think you're right when you mentioned time, because when you think about these errors, they do lead to costly investigations. We do take a lot of time to resolve, and also they could lead to delays in your manufacturing scheduling. So those are costly time issues. But also, once you have a well understanding of your process and you're in full control, your efficiency improves. And some of those time savings that you're getting from, not having to allocate as much labor and personnel into the particular unit operation, that could convert into actual savings as well. So there are many ways of thinking about how to reduce costs.
Yeah, and I was talking earlier, I mentioned human intervention. So I think that's a key concept, that we're trying to minimize human intervention. It's not that we have anything against humans. It's just that we know from experience that the errors and the variation are coming from when you put the burden on your technician to make things work. I think you're just asking for issues. So we can avoid that by designing, the process so that it doesn't involve so much judgment and intervention.
Mm-hmm.
And speaking of that intervention, we've had several situations where our clients had difficulties getting their powder cell culture media into solution. Could you give us just a bit of insight into ways for improving media solubility?
Well, right. So I guess we've mentioned a few, but for example, substitution, more friendly, more soluble components, more stable components. We could also do things like separation of components that don't get along with each other, cause problems because of interaction. We've even talked about what I might call complexation. So complexing, metals with chelators, or where they can associate with something that will stabilize them or perhaps maintain the proper valence. So those are just kind of a list of some ideas. I don't know, Mike, you want to add to that?
Yeah, I mean, most of these ideas were understood, right? We are basically just taking that understanding that it's well known around the industry, and just applying it to our media. We know that certain components are more soluble in the salt form, and that allows us to also prevent or not use those pH titrations that are typically needed to get those in solution. So we know how to improve media, media component solubility, but also there's also, like you mentioned, we could start looking at chelating different components and seeing whether or not this could actually improve their solubility.
So, to summarize, based on these many discussions that we've had with folks in the biomanufacturing space, what would you say to them? What is the real value that we can provide to them for their processes with the MSAT service?
Well, I guess anytime we talk about what we're doing in terms of cost savings or even reducing errors, I mean, you don't really have to convince anybody that that's valuable. I think everybody's looking for ways to reduce risk, to reduce cost, and I guess we didn't really elaborate on why reducing cost is such a popular subject these days. I mean, it's always important, but these days, as our industry matures, I think that subject of cost is becoming more and more important, and it has to do with things like biosimilars. It has to do with the global supply of pharmaceuticals and vaccines. I mean, that's fresh in our minds. We're looking for ways to reduce cost and reduce risk. And I would say this program is squarely focused on very effective ways to do both. So I guess, again, I don't feel like we need to, typically we don't really need to persuade people about how important this subject is. I think sometimes it gets underestimated because, it's not rocket science, so to speak. It's really focused on some very practical issues.
Yeah, I agree with you that, most people don't really think about this, in operation. We generally think of media preparation as something that is easy to do and should be no problems. And it's not an issue until there is an issue. Something comes up in your process that starts, giving you, scheduling issues, or, you're not able to meet your supply of these valuable products to the patient. So we really want you to have a really deep understanding of that unit operation, so that you feel like you're always in control, and that you're able to predict whether the performance of that media will be always reliable. So you have you could build models with the different factors that are present in the preparation method, so that you have that deep understanding of where all these factors are affecting your performance, as well as we are now utilizing some more in silico computational methods to be able to predict the stability of media. In this way, we're able to quickly understand how these multiple components are interacting in silico, and then test those hypotheses in the bench. So with all these tools, we're able to really give you a complete package where you have full confidence in the preparation method, so that it is always reliable and your facility operates at its maximum efficiency.
And even if you transfer from site to site, like I talked about global supply, so that's another thing that's important, like making it more of an engineered solution instead of depending so much on your technicians. I think that's always a good idea.
Yeah.
Thank you, Tom and Mike, for those additional thoughts that you had. I believe we had a bit of a glitch there. So if you could just summarize for everybody here listening in, based on the discussions we've had with all of our clients in biomanufacturing, in bioproduction, in all these spaces, what do you think the real value is that the MSAT service brings to our clients?
Yeah, okay. Well, as I mentioned, Borga, I mean, this service was not our idea. So really, it was the client's idea. They were asking for help. We found a way to meet that effectively, and in a way that addresses the needs of many clients. And just again, to repeat, that the needs are basically practical needs in terms of improving the reliability or reducing the cost, reducing the time, improving efficiency. So instead of being, sometimes I call it rocket science. It's not like a highly technical issue, but it is actually quite important. And Again, it's something that addresses the late stage needs. So I think the value is fairly obvious. Again, the fact that the clients were looking for help on these subjects is what really validates, the relevance of the service to me, and the fact that we've been successful in a number of case studies as we shared. So I think that's, to me, that's what demonstrates the value, and we've been quite happy to see how many people are interested and how many people are having the same problem. Sometimes I'm amazed because it's almost like word doesn't get around that you don't have to follow these, kind of bench scale practices when you scale up. There's a difference between bench scale practices and then large scale bioprocessing. So I'm sometimes surprised that people are still doing kind of old-fashioned things.
Thank you, Tom. A question that has come in from our viewers: what basic strategy could I follow for improving cell culture media preparation to ensure the robustness of my process?
Well, that's kind of a broad question, but I mean, some of the concepts that we described in the webinar probably would help to answer this. I think, the idea of eliminating adjustments, I think that's a good place to start. So that's what when I talk about the bench scale practices, it's common practice at bench scale to, do titrations or QSing up to a volumetric, mark on a volumetric piece of glassware or something like that. But I have to laugh a little bit because when you're at large scale, when you're at large volumes, QSing and adjustments, those are just things that are not necessary. So, that's where I would start when I talk about a basic strategy. I'm not sure if that's what the question is about, but, that whole, subject I just brought up is thinking more practically about the technician. Maybe it's looking at it from the point of view of the technician instead of, the bench scientist. Maybe it's as simple as that. I don't know, Mike, what do you think about a strategy?
Yeah, definitely. There are definitely different ways of approaching these issues. And calling it basic strategy is a good way because, these are simple things that we do to simplify the method, right? What we're basically trying to do is make it a more reliable process that you can every time be consistently hitting those targeted set points without having to do those adjustments. So yeah, we definitely have very basic things that we work on, but once you go deeper into them, they're not so basic, right? They involve knowing a lot about the chemistry that's going on, the interactions that are going on, and how to avoid certain interactions that we know are going to be, bad for the consistency and the reliability of this media. So we definitely know what those different issues are, and we have identified them and identified simple solutions through the understanding of that basic chemistry. So yeah, it sounds pretty basic to do these things, but that's why we're actually trying to provide it to the customer as a service, because we feel that this knowledge that we've gained, that Irvine Scientific has gained over the last 50 years, is something that is really valuable for everyone out there.
Thank you, Mike. Another attendee is asking, would my formula have to be changed significantly in order to improve solubility issues during the preparation process? Because I do want to keep my same formula.
Okay, yeah, we hear that a lot. Actually, just to clarify, the final formula of media, is not necessarily changed at all. So actually, most of the projects we have, we're constrained by that requirement that nothing changes. The same chemistry, the same nutrient concentrations, the same pH value, everything's the same at the end. This is more about the methods of preparation. And so that's where we do like to engineer the formula so that the preparation can be made more reliable, easier, more efficient, things like that. So basically, don't worry about the change to the formula other than the fact that, your methods will change. Now, that alone, I realize, presents some challenges because, you might need to order something as a new part number. And, that be a challenge all by itself, and you might need to validate the new method. But in the end, if you're interested in having those benefits, improved efficiency, reduced cost, improved reliability, I think those are some pretty valuable offerings. And so it's worth, say, changing your methods, changing the part numbers of things. But I would assure you that we can achieve the same chemistry. That's usually what, we're being asked to do.
Right. I think in terms of the chemistry, we try to always end up with the same formula, basically. And it definitely depends on the flexibility of a client, at what stage they're in, how set they are into that formula. And if basically there's no way to change absolutely nothing into the formula, then we start looking at the method itself and trying to see if there's ways to simplify that method without changing the formula, because we know that sometimes that's not possible. So it all depends on our clients and how much changes we can make to that formula, but we definitely like to focus more on the preparation methods and simplifying that so that, like we said, it's going to be consistent and it's going to be, with minimal amount of risk in terms of the errors that could be made while preparing that.
And then I think we have time for one last question. Does the MSAT service help with the physical testing of new formulas, or do you provide just a formula on paper?
Okay, yeah, so that's gives us a chance to illustrate that we're really not just, giving you a piece of paper, or I guess that's an old-fashioned term these days, just like a written, instructions or formula. We really are also involved in, verifying the results, or sometimes it's really just diagnosing what the challenge is, because, testing is definitely within scope of what we do, because we're trying to understand. And Mike, I think you had a case study related to this where we were actually looking at some of the osmolality values, pH values, and other factors in the preparation process and how they were impacting the preparation process.
Yeah, I mean, at the end, we could have all kinds of hypotheses and theories of what's going on, but we have to prepare those prototypes in order to test them, right? We don't just tell you what we think on paper. We actually will build those prototypes, make the media that you need to test on your site. So you'll have, either the powder or the liquid media. We'll prepare for you, ship it out, and you could do the testing on site, and then we'll help you with the analysis of those results.
That's right. So I recall, sorry to interrupt, but I think it was, they were wondering if the method of preparation was impacting their cell culture performance. So we needed to send the samples to them so they could actually do the cell culture testing.
Right.
We don't have the cells, so they were the best place to evaluate that. And that was a great collaboration.
Right. And we'll build that DOE for them and build those different runs or prototypes that are needed for that run. This is basically the full service that we provide.
That's great. Thank you so much, both Tom and Mike, for giving us these overviews this morning. Thanks to everyone who joined us to hear all about the MSAT service. I encourage you to visit our website at FUJIFILM Irvine Scientific and check out the MSAT service and, pose any questions you might have there too. And thank you. Everyone have a great day.
Thank you. Bye.